We talk about aging like it's a clock on the wall. Tick, tick, bad knees, reading glasses, a weird noise when you stand up. But biologically, the more interesting target is not time itself, it's damage. And one of the more suspicious little characters in that story is the senescent cell. A senescent cell is basically a damaged cell that has stopped dividing. That part can be protective. If a cell is stressed or battered up, not dividing anymore can reduce the chance it turns cancerous. Fair enough. Good instinct. The trouble is, some of these cells don't just retire quietly to the veranda. They linger. And while they linger, they secrete a stew of inflammatory signals and tissue-altering molecules. That mix is often called the SASP, short for senescence-associated secretory phenotype. Bit of a mouthful. In plain English, cranky old cells sending out toxic group texts to the neighborhood. Those signals can stir up inflammation, distort how nearby cells behave, and generally make tissue function worse. So the target here isn't immortality in a silver bottle, it's selective cleanup. Less biological clutter, less inflammatory static, better odds the body can keep its footing. Now, into that picture comes something called SenoVax. And very important, this is how it is described, not a settled fact of medicine. SenoVax is presented as a senolytic immunotherapy. Senolytic means aimed at clearing senescent cells. Immunotherapy means using the immune system as the tool. The idea, as described, is to train immune responses against markers associated with senescent cells, so the body becomes better at recognizing and removing them. Sort of like giving the bouncer a clearer guest list. Terrible analogy, actually. Let me try again. It's more like cleaning your shed by finally labeling the boxes properly, so the obvious junk doesn't get left there for 10 years. I say that as a bloke literally recording near a shed. The episode is grounded in reported March 2026 mouse results. Mouse results. Not human life extension, not proof that aging has been solved. Because if anyone says that, mate, put your wallet back in your pocket. The model mentioned here is doxorubicin-induced systemic senescence. Doxorubicin is a chemotherapy drug, and in research it can be used to trigger widespread cellular damage and senescence in mice. That makes it a stress model, not the same thing as ordinary human aging unfolding over decades. And that distinction matters because there are two words people love to blur: lifespan and health span. Lifespan is how long you live. Health span is how long you stay relatively functional and well. You can improve one without fully transforming the other. So when we hear about these mouse data, the smart move is not to ask, have they beaten aging? The smart move is to ask, what exactly improved, in what model, by how much, and does any of that plausibly carry into humans? That's where the real intrigue lives. Here's the elegant bit, and it's elegant in the slightly sinister velvet curtain sense of the word. If senescent cells are pumping out SASP-driven inflammation, then clearing them may do more than tidy up debris. It may change the whole tissue environment. Less inflammatory noise, less collateral stress on neighboring cells, more room, in theory, for repair programs to work without being constantly sabotaged. That logic is why a pairing strategy gets attention. One arm clears, the other arm repairs. Sweep the workshop floor, then bring in the mechanic. If you skip the cleanup, you're trying to rebuild an engine while someone keeps flinging sand into the carburetor. Old bike reference, sorry, couldn't help myself. The repair side, in this reported setup, is described as StemCellRevivify. Again, described as, not clinically proven as. It's presented as a personalized mesenchymal stem cell platform intended to restore regenerative capacity after damage. Mesenchymal stem cells are a type of cell often studied for their ability to support repair and modulate inflammation. People sometimes picture stem cells as magical shapeshifters that become anything. That's too loose. In many research contexts, their importance may be as much about the signals they release and the environment they influence as about directly replacing tissue. Biology loves subtlety. It never just kicks the front door in. So the strategy, as reported, is first, reduce the burden of senescent cells and the SASP mess they create. Then, support recovery with a regenerative platform. That is a coherent preclinical idea. Coherent does not mean confirmed, but it's not nonsense either. The reported mouse outcomes are the sort of things that make scientists lean forward without yet throwing confetti. Improved survival was reported. Physical performance improved as well. And there were improvements in organ-related markers. Now, markers is one of those words that sounds sturdier than it is. A marker is a measurement associated with a biological state. Useful, yes. Final proof? No. A better marker can suggest healthier tissue function, lower damage, or reduced inflammation, but it doesn't automatically mean a mouse is biologically younger in every meaningful sense. Same with survival. In a doxorubicin-induced systemic senescence model, better survival is notable. But it's still survival in that specific injury and stress setup. It does not equal demonstrated extension of normal lifespan in naturally aged animals, and it definitely does not equal human longevity extension. I know, that caution sounds like a wet blanket at a barbecue, but in this field, caution is not the enemy of excitement. It's the lock on the medicine cabinet. Still, if the combination really did improve survival, physical performance, and organ-linked readouts in mice, that suggests something worth following. Not because it proves the future has arrived, but because it hints that clearance plus repair may be a sharper move than either one alone. So what do we do with all this? We hold two ideas at once, like carrying fuel in one hand and a match in the other, and deciding wisely not to be an idiot. First, these company-announced preclinical findings could point toward a genuinely interesting therapeutic direction. Second, they are not peer-reviewed proof that human aging can be reversed, or that human longevity has been extended. Those are very different categories of evidence. Preclinical means before human clinical proof. Often that means cell studies, animal studies, or both. Valuable, absolutely. But biology has a long and slightly humiliating history of looking brilliant in mice and then fizzling in humans. Different lifespans, different immune systems, different disease contexts, different everything, really. If senolytic immune therapies like the one described here do translate, the implications could be broad. You'd think about cancer care contexts where treatment itself can drive senescence and long-term tissue damage. You'd think about frailty, where accumulated dysfunction chips away at strength and resilience. You'd think about organ decline, where inflammatory wear and tear may help push systems off balance. And then there's multimorbidity, which is the charming medical term for having multiple chronic conditions at once. Aging rarely arrives as one neat villain. It comes as a syndicate. But could is doing a lot of work there. We do not yet know whether targeting markers associated with senescent cells will be specific enough in humans, durable enough, or safe enough. We do not know how broad the benefits would be, or whether some tissues respond while others stubbornly refuse. We do not know whether repeated treatment would be needed, whether immune training would wane, or whether unintended effects would emerge. In aging biology, the unknowns are not decorative. They are the furniture. So the next gates of evidence are pretty clear. Naturally aged mouse studies would matter a lot more than injury-induced models alone, because they get closer to ordinary aging biology. Then clinical trials in humans. Then the hard questions inside those trials. Safety, durability, reproducibility. Safety means, are we causing harm? Durability means, do effects last? Reproducibility means, can other teams see the same thing, not just the original group? If a result only lives in one lab announcement, it's a rumor wearing a lab coat. That's the state of play. Not the fountain of youth, not snake oil by definition either, just a sharp, intriguing preclinical strategy aimed at one of aging's nastier cellular leftovers. We'll keep an eye on the evidence as it comes in, nice and steady, no fireworks just yet. And next time, we'll poke at another corner of the longevity world and see which claims survive the torchlight.